Hello, everyone. It's Dr. Andrew Heyman. Happy to be back with you to talk about the next layer of CIRS, which is our transcriptomics research. I think this is going to be something that is interesting to you and even exciting in the way that advanced science is coming together with real world clinical problems. And to me, this is sort of the coming attractions of where a lot of medicine is going. So recall, we're looking at this notion of an exposure that ignites a genomic response, and we're able to measure that by measuring mRNA. And mRNA, of course, are the signaling molecules that reflect the DNA and then go to the ribosome that are assembled there to produce proteins. Proteins are then folded and glommed together to create small molecules. Our work is in transcriptomics, so we have the capacity to perform research on our study subjects, looking at the patterns of gene expression as it relates to this topic. We've been collecting this data since 2015, and now we have a relatively large data set where we've been able to characterize hallmark patterns of gene expression that underlie this illness and also give greater insight into what's really going on in this patient population. It's an extraordinary breakthrough in that regard, and it's required meticulous research for a number of years to come to some reliable conclusions on the ways in which genes lose regulation and begin to behave in maladaptive patterns that drive the illness complex of the CIRS patient. And the way I explain it to patients is that we have about 30,000 genes in our genome. And with that being said, in the field of molecular biology, with the study of genetics, and I want to make a distinction between genetics and genomics. Genetics is, of course, the capacity to measure the structure of the DNA that's been inherited. And imagine if these 30,000 genes that are present are like personal instruments in your own symphony orchestra. So the analogy would be that in the study of genetics, we would be evaluating to understand if each of the instruments were built properly. And this is to say that maybe a clarinet doesn't have all the right keys, or the cello has strings in the wrong order, or the slide on a trombone sticks. The inference is that if there's some sort of physical alteration to the instrument, that the sound that it makes not only might be altered but unpleasant or doesn't fit in to the orchestral arrangement. This is the same for genetics, this notion that we can measure SNPs and variations to determine if those physical alterations might confer some sort of abnormal cell activity. But it is an inference. And I think that, the world has really moved on in terms of understanding that, would one instrument in the setting of 30,000, even if it wasn't built well, could it really profoundly influence human health overall? Sometimes yes, most of the time no. What we're learning is that as we frame shift to ask the question, okay, that's one idea that maybe an instrument wasn't built well. But the other idea is, what about whole sections of your orchestra? What if your brass section was playing too softly, or your percussions were out of time, or your woodwinds were too loud? That's genomics. That's this notion that as we group genes together and look at what their pattern of expression is, what the music that they're playing is showing us, we can determine whether or not there's melody and harmony, or is there noise, and does that noise over time confer illness? And I think the answer what that we're finding is yes, that it's how genes sort of group together and either pattern out normal expression or not that really matters. And so this notion that we're moving from genetics to genomics, from structure to function, from how your instruments are built to the music that they're playing, that's what we're looking at with transcriptomics, and we're able to determine what our genes are doing activity wise by measuring the amount of RNA that's produced. So this transcriptomics is becoming a very sensitive indicator of both disease status and emerging health hazards. Now, recall how we did this in an aggregate population. We knew that there were abnormal patterns of gene expression that were present even pre-VIP, after appropriate therapy, and then we offered VIP and we saw a change in that pattern. Well, in addition to that, we can go a layer deeper, not just asking the question, does this patient have patterns that correlate with CIRS overall? But we also have research to show that there are very specific patterns of gene expression for active Lyme. There's a different pattern for treated Lyme. All of this was published in 2016 out of Johns Hopkins. They did some incredible research to show that there was a reliable pattern that emerged within the mitochondrial DNA as a result of an active infection. Tremendous research, very complicated, high mathematics and statistical analyses, but they broke the code and they figured out the genes that are present that turn on in the setting of tick-borne illnesses. Um, in addition to that, it was our research group that identified the genes, Mapkinases, that turn on in the setting of a mycotoxin exposure. And so this was a real breakthrough for us. So we could tell if a patient was reacting to their environment. But interestingly, in our last data set of 1,000 subjects, only about 7% of them were showing a reaction to mycotoxins. And I'll get into, well, what else do people react to? Um, we can even tell whether or not Marcons and that paletoxin is affecting the mitoribosome because we can see suppression of the mitoribosome in our genomics. And then also, should I test for actinomycetes and endotoxin? You might ask, what is actinomycetes? Well, actinomycetes is a bacteria that also grows in the home and it makes people sick. It releases toxins and an endotoxin into the air. And when patients breathe it in, they can become very ill, just in the same way that mold can make people sick. Interestingly, in that same data set of 1,000 subjects, if only 7% of them were showing reactions to mycotoxins, 42% were showing a reaction to the actinomycetes, a six-fold increase. For those of you who really thought that a water damaged building causes illness, you're right. For those of you who thought that the pathogen of choice was mold, you're wrong. It turns out that actinomycetes is probably the culprit of choice. And over time, our data set became more and more convincing of this idea. Now, you still remediate the bacteria in similar ways to how you deal with mold, um, but it's not the same. And it grows under different conditions than mold. It doesn't need as much water as mold. It just needs some dust and certain building material. But if you look at Dr. Sulyak's work out of Europe, Denmark, Finland, they're doing tremendous research to show that actinomycetes basically grows in most living spaces, and many people are sick from this bacteria. So if you've been chasing Lyme and you've been chasing mold, you've been chasing shadows. Most of the time, it turns out when we measure gene expression patterns, it's from something else altogether. And this data is only about 18 months old. So if you haven't heard of this, I'm not surprised. Um, does necroptosis appear? Yeah, about 1/3. So that means endogenous sources of inflammation, meaning genes are turning on that drive the inflammatory process. Do I have cytokine activation? We can measure this. Do I have upregulation of histamine genes? Now, for those of you that have sort of fallen into the mast cell trap, what does that mean? It means about 60% of our patient population does show a histamine problem, but it's because of histamine gene activation, not because of mast cell issues. So every nucleated cell in the body is a problem. It becomes an engine of histamine, and all the tissues fill up with histamine. It's true, histamine is an issue. I don't fully buy into the mast cell story. We can also see gene activation for PTSD. There's a very strong relationship between a biotoxin process and an abnormal stress response. And then, what is this vascular hypothesis of neuronal injury? I'll get into that. It means that the brain really is at risk in this illness. If I'm going to use VIP, what dose do I use? We'll talk about that. Methylation and demethylation. Methylation, demethylation are not on the worry list. We know they don't play a big role in CIRS. Are my T cell synapses with dendritic cells working? I'll show you that too. This is a very important feature of the illness. It helps us to stage progress. Do I have a platelet problem manifested by tubulin activation? This is another issue that puts the brain at risk that I'll get into. And then, am I going to hemorrhage if I'm exposed to a water damaged building? And I'll show you that as well. So, am I incurable? And the answer is under most circumstances, no. That transcriptomics brings this concrete hope that through validated testing and measurement and proper treatment, we can bring patients back to a normal state of affairs. We can cure this illness. We can reduce their inflammation. We can change their cell responses by re regulating expression of DNA, and we can induce cure. So we've done this by using RNA-seq, which is our ability to measure mRNA production, and it's an incredible insight into what's going on with these patients. Now, one of the aggregate activities that we've been able to identify is staging patients based on their step of therapy overall, meaning stage one is a pattern of gene expression overall in the mitochondria that indicates no viable treatment has occurred. That doesn't mean no treatment has occurred. It just means that the genes are expressing in a way that this is a fully expressed CIRS patient, and the genes have not changed whatsoever, even despite maybe some therapy. Stage two, we can tell they're going down the treatment path and genes are starting to turn off. And that is validation that the earlier steps in treatment prior to VIP do work, and there's some genes that respond. But by stage three, they're on VIP or after VIP, and we can tell that VIP has been working and they're turning off those residual genes. And stage four is they're cured. So, this is how we understand what's going on with these patients relative to the overall success of therapy. So I can tell, are my patients getting better? Where are they, kind of overall in their stage of therapy? Have I gotten them to cure or not? That's a tremendous amount of information already to say I can tell where they're at in their treatment progress based on the overall pattern of gene expression. So the N originally was 161 subjects, which is a pretty good number. We're now, well over 1,000. And genomic research has high sensitivity and specificity, so we don't need large data sets to derive meaning out of our cases. So, what does this look like? What when we get a genomic report, what does it first appear sort of from an aesthetic perspective, and then also what are the areas that we're measuring? So, I've pulled segments of a genomic report to demonstrate exactly what I was just talking about. So, we're able to ask the question number one: is the patient a CIRS patient? And so we aggregate together a variety of genes that, if three or more are upregulated, it means that yes, we're able to easily confirm the diagnosis of CIRS. And you'll notice that there are circles, and some of the circles have colors, and all the circles have numbers. So a circle is a gene, and there's a number associated with each gene, which determines its level of activity. And then we color code that if it's overly expressed or underexpressed. Overexpression is a light orange all the way out to red, and an underexpression is a light blue all the way out to purple. And this notion then that here is the music that's being played, and we have a section of the mitochondrial genome that's too loud and others that are being suppressed. But in our criteria, it's overexpression that matters. So if three or more in section 13 and 14 are overly expressed, it's a patient who has CIRS. And so you can see this patient obviously falls into that category. So we're able to ask the broad question, are they a CIRS patient? Don't forget that what we're doing is we're sort of characterizing patients based on their mitochondrial genomic expression, looking at factors of inflammation, factors of abnormal metabolism. Recall that they push into aerobic glycolysis. They're moving away from an energy plant and into a battleship. And under that state of affairs, Their energy efficiency is downregulated. They're becoming glucose dependent, which means aerobic glycolysis and proliferative physiology. But yes, they have an inflammatory component too. So we're really kind of in this mixture of CDR1 and CDR2. How do I know that? Well, I know that because I'm able to measure hypometabolism or aerobic glycolysis revealed. So here's aerobic glycolysis in CDR2 that Dr. Navio mentioned from a small molecule perspective. What we're showing are the genes that are downregulated and force the cell into hypometabolism. They're moving away from oxidative phosphorylation. And if you notice in stage one, it's 80% of patients are in this hypometabolic state. As we treat them, and we do all those prior steps of therapy pre-VIP, look at this. Their hypometabolism improves. So we can tell that they're moving down the path of therapy. And look at stage three. Once we get them on VIP, miraculously, they go to normal. All of them go to normal, which is amazing. In stage four, basically, it's all eliminated. So this notion of aerobic glycolysis or proliferative physiology is all over the metabolic literature, and it determines that the cells are becoming glucose dependent, and therefore the patient is insulin resistant and gaining weight. Even worse, we know that the BDAC, which are membrane transporters on the outer cell membrane, can be closed and worsen this process with azole antifungals. And I would urge you that at least in a CIRS patient, don't use the itraconazole and fluconazole because they will make this process worse. Okay. We've also found that, and this is profound, that when patients are in this hypometabolic state, plus an over or upregulation of the insulin receptor substrate genes, that this is what causes the damage to the brain on neuroquant, not inflammation. It's the hypometabolism, it's the aerobic glycolysis, it's the dependency on glucose that the neurons in the brain do not like. And we start to see areas of interstitial edema as well as shrinkage, which is the atrophy or the dendritic pruning that I've lectured about. So this notion that neuroquant can pick up on these changes in the brain, it's actually driven by the abnormal metabolism. We also know that when you pair these two elements, the hypometabolism plus the upregulated insulin receptor substrates, that this also drives an anion gap and also is what's responsible for pulmonary hypertension. In these patients, you get vasoconstriction of the pulmonary artery, ballooning of the right atrium, stretch of the sinoatrial node, tricuspid regurg, all as a result. So there's a cardiopulmonary impairment that's occurring because of this combination of metabolic findings, as well as suppression of T regulatory cells. How do I know this? Well, here we go. Here's a typical patient who all of the metabolism genes that control ATP production are downregulated. And so this is a classic presentation of an individual who's hypometabolic and they have upregulation of those insulin receptor substrate 2 genes. This is the worst position to be in. They're in the most extreme. They're a stage 1 patient that has not been treated at all. So how do we know this? They're going to have a widened anion gap. They're going to be at a greater risk for changes in the brain. And that when we look at these patients, we can map out worst to least depending on the position of the hypometabolism and the insulin receptor. So, what you want to see over time with treatment is clearing out of both of these sections so they look like they've gone to normal. And in this patient, they are pushed into aerobic glycolysis because of the patterns of gene expression. They're not burning oxygen, they're burning glucose. It puts the brain at risk, it puts the cardiopulmonary system at risk. It leads to a metabolic acidosis, so they're not ready to detoxify. In addition to that, this is what drives fatigue and brain fog. So, all of this is occurring as an abnormal pattern of gene expression driving problems in metabolism. And by virtue of that, this notion that this is a solely inflammatory process is incorrect. We now understand that through our genomics research that these patients are in fact having issues with a metabolic efficiency as much as being in a pro-inflammatory state. Now recall I also mentioned T cell receptors. This is another hallmark. As much as hypometabolism and aerobic glycolysis is, so too is the downregulation of the genes that control the T cell receptor genes. So this notion that the immune system is not only inflamed on one hand, but inefficient on the other because another hallmark for CIRS. What does this mean? Well, recall the idea of antigen presenting cells that are inefficient because of the presence of HLA. Remember that this is about 9 genes on chromosome 6 that confer this poor antigen presentation. Well, what is the antigen presenting cell presenting to? What is it showing the epitope for? Well, it's showing it to a T cell receptor. Well, guess what? Now we know there's an acquired deficiency on the adaptive side of the immune system, so that there's a further downregulation and a further inability to produce antibodies as T cells aren't able to trigger B cells efficiently. So there's this inherited inefficiency as well as an acquired one as part of the bridge between the innate and the adaptive side of the immune system. This is also how we stage patients to know, are they in molecular hypometabolism? Do they have downregulated T cell receptors? And for those of you that are sort of concerned about whether or not these patients are ready for a COVID vaccine, they're not, because if you give them a vaccine, they're not going to be able to respond to the antigen very well and turn on an antibody response. So, what I caution my CIRS patients around is wait until I get you down the path of therapy, at least on VIP. I know I'm turning on the T cell receptor genes. Now you'll be ready for a vaccine. Prior to that, they have a very inefficient adaptive immune response, and they're not going to respond to vaccines very efficiently. And look at this. We know, for example, that basically these receptor genes don't do anything with any stage of therapy until VIP. Look at the dramatic drop in the CD3D once we get them on VIP. That's incredible. So this is part of that irreducible nugget of it's going to remain negative until I get them on VIP. And the same is true for the CD48. Again, a precipitous drop. We get a little bit of progress on early step, but not until we get on VIP do we see a real change. Now, how do I know that a patient is in a water damaged building? Well, we found the Mapkinases that turn on in the setting of a mycotoxin exposure. But remember, I said this is only 7% of our study subjects. It turns out, there's a sixfold increase when individuals are reacting to the bacteria in their environment called Actinomycetes. If you haven't heard of this before, don't feel bad. This is all new research, but it is all over the literature. And we're the first research group to show in genomic fashion the degree to which humans actually negatively respond to the presence of these microbes. What this is also saying is that number one, homes are not sterile ever. Number two, it's more that the home is developing a dysbiosis because of amplified microbial growth, as opposed to going from a non sterile environment to a sterile, because remediation, we now know, doesn't really get rid of a lot of this stuff. All it does is hopefully create eubiosis and a better balance of the microbes so the people who are inhabiting the home don't become as sick. So we can tell based on the patterns of MAP kinase expression whether or not the individual is sick from mold, sick from bacteria, sick from the endotoxins the bacteria release. All three, or a combination of those three. This is what it looks like when a person is sick from the bacteria and the endotoxins, which is the CD14 and the toll-like receptor 4 genes. Amazing. So we can tell that this individual is not sick from mold. They're actually sick from the bacteria and the endotoxin. That determines what we swab for. It determines how we remediate. So note this. So here's our 7% in the mycotoxin. Here's the 42% in the actinomycetes. And the endotoxins, people are reacting around the 30% level. And if you notice, again, they don't really respond until we get to VIP. We don't turn off these genes until we get to VIP. We just published a new study in Medical Research Archives. This is the first summary research paper on actinomycetes and its role in water damage building. So this is about a 35-page paper that goes into great detail on this whole topic of actinomycetes. And we review the literature, we summarize our own research, and we talk about remediation methods too, to some degree. What about Lyme disease? I realized that there was some interest in Lyme. We're finding that, it's not as common as people think, mostly because now we have better testing methods. Thank goodness Bouquet in 2016 at Johns Hopkins found the genes that turn on for Lyme disease, and they also found the pattern of gene expression for properly treated Lyme. They're different, and it's actually based on a calculation that I've given you. It's an average, and we take four genes, and if their average is above 1.37, they have Lyme. And if we take the average of all of the above genes except the EIF4G2, then we know that they have properly treated Lyme. 6 months after treatment, they tend to go back to normal. The genes go back to normal on their own. I tend to use just doxycycline. There are some herbs that I use as well. I'm finding Lyme is not the hard thing to treat. I realize there's all this paranoia around Lyme. Now I have a verification to show whether or not they're actually in a properly treated Lyme fashion. Usually patients respond well just to 3 or 4 weeks of doxycycline. I know that's not very exciting, and there was a lot of interest here, but the reality is I can tell if my patients have recovered from Lyme. Almost all do from a little bit of antibiotics. It's not the boogeyman that people make it out to be. The problem is most of the quote Lyme community will not take into account all of the knock-on effects that Lyme induces, all of the inflammatory problems, the metabolic derangement, the genomic findings. You have to fix all of that for patients to get better. It's not enough just to eliminate the infection. You have to go through the whole process, the entire treatment protocol, including VIP, to get these Lyme patients back to normal. It's not about killing bugs. It is truly about everything else that comes after that, which is why I've spent so much time going over my treatments of how I take Lyme patients down the path of therapy. What about other infections? So we know that the defensin genes can turn on in the setting of viruses and bacteria. This is wonderful because Epstein-Barr virus, HHV6, these are all DNA-based viruses that intercalate into our own DNA. And we often have positive serum markers, and patients can mistakenly think that their symptoms are related to an ongoing viral process. But now I can correlate that to patterns of gene expression in the defensin category. And if they're overly expressed, I know they're dealing with a chronic infection, not Lyme related, but usually due to viruses or some bacteria, and then we work to sort of stamp those out. So now I have a check against the blood markers, and oftentimes they're negative, but sometimes they're positive. This was a positive patient of mine. In addition to that, here's where we see some success of functional medicine. RIPK1 is one of the major inflammatory genes. So when it's turned on, we have a lot of cytokine production. And notice that just through the early stages of therapy, how this gene turns off, and we're very good at reducing inflammation, but we don't get that full metabolic shift until we do VIP. Now, I also briefly mentioned the vascular hypothesis of neuronal injury. Well, what is that? Well, look at these genes. These are coagulation genes, and they're responsible for microclots. And what we've learned is that if three or more of these genes express, then microclots will start to form in the small capillaries that feed the brain, and it creates poor blood flow into the central nervous system, which stimulates beta amyloid production because of the presence of the coagulation products. This induces microclots in those small capillaries and distal hypoxia that leads to tau formation. Microbleeds are common as a result of well. So if these genes are not silenced, this leads to neuronal death. And we've correlated this in particular with vascular dementia. So now we understand the link between an exposure, microclot formation, beta amyloid plaque production, tau phosphorylation, and precipitation of a vascular dementia. This is the first time that gap in the research has been filled. We never fully appreciated the degree to which microclots could lead to a very common form of dementia. And thank goodness we have VIP to turn these genes off. And notice that a lot of these genes remain active until we give VIP. And we also know that in terms of damage to the brain, the cut point is three genes. So at three genes or more, we see a marked leap in the areas of nuclear atrophy or essentially injury to the brain. as a result of hypercoagulation. This is an extraordinary insight that we now know further puts patients' brains at risk. Molecular hypometabolism puts the brain at risk, hypercoagulation puts the brain at further risk. And we know that these genes remain active until we get them on VIP. Notice this, there's no change in gene expression until we get them on VIP. This is why when practitioners would do blood panels looking for clotting factors, they're often elevated. Anticardiolipin antibodies, antiphospholipid antibodies, von Willebrand factor, D-dimer, all the rest are part of this illness complex. And now we know why. And it specifically puts the brain at risk. Even worse, I had mentioned the tubulin genes. So these confer structure to the cytoskeleton. And when these genes are overly expressed, it turns out platelets in particular become floppy. And if you combine that with the hypercoagulation of three or more of those genes that I just mentioned, it puts the brain at significant risk because not only do you get microclot formation in the capillaries, it means that also beyond that, there's even worse blood flow to the brain because of platelets that are getting trapped and compressed in those same areas as well. So there's even worse hypoxia and distal ischemia as a result of tubulin activity. So this combination is what accelerates the vascular dementia risk. What about COVID long haulers? I'm sure you've heard about COVID long haulers. They're CIRS patients. Think about what COVID long haulers talk about: brain fog, mental fatigue, aches and pains, bloating, mood disorders, cognitive decline. Certainly sounds like a CIRS patient. Well, it is a CIRS patient. And we can see that just like HIV, COVID induces abnormal apoptosis as a feature of how it overtakes the machinery of cells. And we can tell that these are COVID long haulers as a result because of the induced abnormal apoptosis. What about mast cell activation? I had mentioned this notion of, yes, these patients have histamine production, but in our case, from what we've seen, we don't believe it's from mast cells. We think it's because one or both of the histamine genes have been abnormally activated. And now every nucleated cell in the body becomes an engine of histamine. So the brain and the other organs and all the tissues do fill up with histamine. Of course, these patients are miserable, and antihistamines can help. Mast cell stabilizers don't really. And that's where I like to use the very high dose diamine oxidase because these patients respond well to that. Now, how do I deal with that? Look, there's no change in their histamine genes until I give VIP. So, again, we can do some symptomatic relief with managing histamine, but it's not until I give the final step of therapy can I turn those genes off. So, what does this mean as a summary? It means that new information of clinical value comes with these novel therapies, and our focus on of remediation is not on the minutia, but it's about sort of restoring normal eubiosis in the living space, which includes actinomycetes and endotoxin, as well as HERTS-2 for mold. What about confirmation of correction of T cell synapse as a target of therapy? Absolutely. So we can see not only improvement in molecular hypometabolism, but also a turning on of those T cell receptors. Recall actinomycetes dominate the transcriptomics. Why are we obsessing over mycotoxins? Better yet, what treatment of a house worked to correct the results? So we're even changing to some degree our thought around what is remediation and what are we trying to do. And then what are some of these other therapies that speed up recovery? Now, again, I use in some ways an expanded toolkit, but actually my patients go down the path of therapy relatively quickly. We can get them on VIP expeditiously. So it is a new day and a new dawn with respect to transcriptomics. Imagine if patients could know if they didn't have, transcriptomics of Lyme. I mean, all these patients that run around thinking they have a tick-borne illness, now we know for certain that they don't. Cost savings would be huge. Imagine if mycotoxin injury were finally ruled out. It is in favor of actinomycetes. Savings on urine tests would be huge. We know that there's a lot of urine testing for mycotoxins, which are not valid, but it also just points in the wrong direction regardless. We know urine tests only reflect the food people have eaten, not the environments in which they live, but it even just, overly suggests mold anyway. What if we knew mast cell activation was not present? Yes, because of histamine gene activation. No more wasted money on therapies that won't help. And in the future, we're really going to focus on insulin receptor recycling, reversal of neuronal injury. The basics of history, physical, and differential diagnosis still are mandatory. So are the visual tests and NeuroQuant, among others. But now we ask, does my differential gene activation need to be corrected? Yeah, there are hallmarks here in CIRS that need to be addressed. And if yes, help is on the way. Now we have a way of sort of characterizing how patients move through steps of therapy, which genes turn off in which order, which symptoms go away in which order, and what's residual pre-VIP and post-VIP. And we have a way of measuring a true cure. And that includes passing the visual test, normalizing blood markers, restoration of a normal neuroquant, as well as resolution of abnormal gene expression. All of that is cure. It's not one or another. It's not just elimination of the mold. It's not just cleaning of the home. It's not even most symptom reduction. We want full cure. And full cure means all the way through the end of the process, including resolution of the abnormal transcriptomics. And recall what that looked like. Remember, this is pre VIP. This is all those other therapies right before we gave VIP. And then look at this beautiful result. Look at how we induce cure. This is Lyme. This is mold. This is other biotoxins. This is the actinomycetes. This is what we now know how to get people back to a regular state of health. It includes removing the exposure, turning off the inflammation, restoring normal metabolism, and ultimately re-regulating gene expression. So this is the biotoxin evaluation process. This is the standard set of therapies that you can find on survivingmold.com. This is my expanded set. This is how we get to full and final resolution. So thanks so much for your attention and time. Hopefully this was a fascinating end to thinking about the biotoxin patient and where medicine is going in the future with the ability of transcriptomics to further segment this patient population, more deeply understand the effects of our treatments, and to demonstrate the possibility of cure.